Hello. Yeah. Okay. So, one second. Let me switch this on. Yeah. So, hi everyone. I'm here for the eighth tutorial of the I Think Biology course offered on NPTEL by Azim Premji University. I am Geetika Agarwal. I'm a PhD scholar at the Center for Ecological Sciences, and I will be taking your tutorial for today. So, first of all, I'll begin with giving you a brief summary of what happened during the lectures of this week, and then we'll have a small Q&A session. And anybody, if there are any doubts, they can ask me in the middle of the session, or later when you see this YouTube video, you can ask the doubts in the comment section. Okay. So, first of all, coming to the contents of this week or what was covered during this week was mostly about species and how do you define species, what is speciation, what is biodiversity, and how do you measure different types of biodiversity, and how do you calculate or quantitatively measure biodiversity of these different types. Okay, so this brings us to the first question of talking a little bit about evolution, since the last week was mostly about evolution and how that happens. So, evolution can be described on two major scales. One is microevolution and one is macroevolution. For microevolution, first of all, you basically, when you're talking about a single species and we're talking about things that are happening within a population, that is microevolution. When you're talking about a small scale, so if you're talking about microevolution, you basically are thinking about how the gene frequency is changing within a population, within a species over time. So when you're talking about how gene frequency is changing over time, but you're considering a single population of a species, that is microevolution. It mostly relies on mutations, that is spontaneous changes in the genes, as we discussed in the last week. Or it's talking about migration, which can also be thought of as gene flow, when individuals are entering or leaving the population, and that is affecting how the gene frequencies or the allele frequencies change within the population. Then you're talking about genetic drift. How within a population, because of certain chance events, the allele frequencies can shift from one way to another based on what events are happening. And talking about natural selection, if the population is under selection for a certain thing, for example, in this image you can see that there is a population of beetles which have two colored morphs. One is green, one is brown. The brown one is very well camouflaged with the bark, but the green one actually pops out and can be picked up very easily by a predator, such as a bird in this case. So that is a natural selection. There's a selection pressure for escaping the predator's effects. So it's better for the beetles to be well camouflaged with the background so that it's difficult for the predators to single them out. So in this little population of beetles, the brown beetles will get selected and they will be able to leave more progeny behind. They're better fit for this habitat. And that makes it easier for them to leave behind more of their gene copies. That is called microevolution, which is happening at a small scale. A simple example is of industrial melanization and peppered moths. If you're not aware of this example, you can go back to the last week's video and look at that, where we discussed about how industrial melanization actually affected the evolution of peppered moths. Then macroevolution, as the term suggests, it's when you're talking about evolution at a bigger scale, which is like above the species level. So if you're taking a zoomed out view of some phylogenetic trees, for example, here is a phylogenetic trees of the wolf populations or the dog populations. So basically, it's gray wolf populations. So if you're talking about, if you take a zoomed out view and you see how the entire family of wolves have evolved over time, then that is something which is called macroevolution, because here you're going beyond the level of species. And this mostly relies on speciation, where one species eventually forms or evolves into two different species. Okay, so if you look at these microevolution processes, mutation, gene flow, genetic drift, and natural selection, if you look at them over longer time scales, for example, billions of years, then that will eventually lead to macroevolution. So this is what micro and macro scales of evolution are all about. And if we take if you look at this flow chart and we're talking about evolution, as we discussed in the last week, there are certain raw materials, like there is variation, which is important. There should be a variation in traits. And there can be raw materials, like there could be a certain mutation, there could be recombination. So this variation can arise because of either mutation or recombination, these two factors. And this variation is actually then acted upon by causal agents like drift or selection. So certain of these traits might get selected, or certain of these traits, just because of drift, might go forward in generations, while the other ones might get erased. And this leads to eventually changes across generations. So as you move further in generations, there will be a change in the variation or in the traits, which is actually evolution. And the consequence of that is that you'll have a population that is modified from the original population, and probably it is more adapted, it is better adapted to the current habitat than the previous or the parent population. Okay, so this is how the flowchart of evolution moves along. And we're talking about what are species, which is the basic question of this week, what we're trying to discuss during this week. What are species? So, um, species, if I simply describe it, it's like a basic unit of classification. For example, like we call the cell, which is the basic functional and structural unit of life. Species here is actually the basic unit of classification. Classification is how we are classifying different organisms that are present on the face of the earth. The species is the basic unit of classification, and it's the lowest taxonomic rank. So all individuals belonging to the same species can actually be thought of as similar individuals, because they can interbreed, and they are genetically more related to each other than two individuals belonging to different species, right? And any organism's scientific name, you might already be aware, when you're writing an organism citing scientific name, it is written as the genus and the species name. So first you write the genus name, and then you write the species name. For example, if you're talking about uh, for example, if you're talking about organisms like foxes, like one of the scientific name is Vulpes vulpes, in which the genus is also Vulpes, and the uh, what do you call it? The species is also Vulpes. For example, if you're talking about the red fox, right? Or if you're talking about birds, then, for example, if you're talking about peacock, then the scientific name of peacock is Pavo cristatus. And Pavo is the genetic name, and cristatus is the species name. So that is a very basic description of how you write down the scientific names. And then you might already be aware about how we classify life. And when you come to classifying life, you have these different levels. First of all, you have life. In life, you have different domains. In these domains, you have these things called kingdom, phylum, class, order, family, genus, and kingdom. Oh, species, sorry. And a small mnemonic to remember this is kids prefer cheese over fresh green spinach, right? So, species is the basic unit of classification. Different closely related species fall into a similar genus. Related genus fall into a family, and they fall into an order, class, and phylum, and kingdom, and so on, right? So that is where kingdom, phylum, class, order, family, genus, species comes from. And there are many different concepts which try and define species in different ways and demarcate these different distinct species from each other. So, some of these concepts that we're going to discuss during this class is the morphological species concept, biological species concept, and the phylogenetic species concept. There are other species concepts as well, but they will not be the focus of the tutorial today, because these are the basics ones that are most popularly used, right? So, we'll come to this later. And but when we talk about species, what happens below the species? So this was what is above the species. There's genus, family, order, right? But within a species there are like varieties. Varieties are commonly used to describe some variation within a species. It could be due to a certain polymorphism or certain environmental changes. A species could have different varieties that are existing, but they still belong to a similar species because they can interbreed and produce fertile offsprings. Then there are subspecies within a species, and these subspecies are used to describe populations which can be recognized because of their distinct physical and genetic characteristics. So these are populations that can actually still be recognized because of their distinct physical and genetic characteristics. For example, there are some bird species, some of them have for example, or there are some butterfly species, some of them might have a white morph, some of them might have a black morph. They might be called as subspecies, but they would still be able to interbreed, and that is why they still belong to the same species. but they can be subspecies because they can be recognized separately. Then there are ecotypes. Ecotypes are actually genetically distinct populations which have adapted to a specific environment. As the term suggests, ecotype. So they are types of organisms that belong to the certain ecosystem. So genetically, they are distinct populations, but they're adapted to specific environments, and they exhibit traits that enhance their fitness to be surviving in that particular habitat. So you're talking about populations that are of a same species, but these populations could be genetically distinct. Like there could be certain genetic traits or signatures in them that make them distinct from each other, and they're adapted to certain specific environments, and they have traits also which make them better fit to be living in that particular environment, in that particular habitat. So these are called ecotypes. So there are varieties which depend on certain variations that exist within a species, could be polymorphisms or could be certain environmental changes. And then there are subspecies which are used for populations which can be recognized because of their physical and genetic characteristics. Then there are ecotypes, which are distinct because they occupy distinct habitats, but they would still be genetically closely related. And then when we're talking about species in agricultural terms, there are these two terms called accessions and cultivars. Cultivars are basically a short form for cultivated variants. So when you cultivate different varieties of a species, they're actually called cultivars. So if a distinct, uniquely identifiable sample of seeds. So an accession is actually a distinct and a uniquely identifiable sample of seeds which represents a cultivar or a breeding line or a population, and can be maintained also in storage for later use for agriculture, right? So it's a distinct and uniquely identifiable sample of seeds which represents a cultivar. Okay. Okay. So moving on, when we discussed what comes below species, so as we were discussing, there are these different species concepts. What we will be discussing today is the biological species concept, the morphological species concept, and the something which is called the evolution species concept or the phylogenetic species concept. Okay. So, but there are these different species concepts. For example, the biological species concepts, which basically talks about reproductive isolation, and it is not necessarily easy to apply, and we will see why that is so in the upcoming slides. But the biological species concept, as we said, it talks about reproductive isolation. So individuals belonging to a similar species, to the same species, can actually interbreed and produce fertile offsprings. But if they are reproductively isolated, if they cannot interbreed and produce fertile offspring, then they're thought to be belonging to different species. Then there is a morphological species concept, in which in which the scientists were actually trying to distinguish between different species based on their morphology, based on their physical characteristics. And this is most commonly applied to fossils or to extinct species, because you do not have access to their reproductive behavior, so you cannot apply biological species concepts to them. And to apply genetic differences also, it is a little bit more difficult. So morphological species concept is most commonly applied to the fossils, or to species that are so distinct to each other that they can be morphologically distinguished. But it also comes with its own caveats. Then there is this recognition species concept, which actually depends on behavior of the species and on mate recognition. So if two individuals belong to the same species, they could actually be able to recognize each other as mates if they are say belong to the different sexes as well. So this is what comes to the recognition species concept. If there is a male and a female and they actually belong to different species, they will not be able to recognize each other as potential mates. But if they belong to the same species, they'll be able to recognize each other as potential mates. Then there is something called the cohesion species concept, where we talk about persistence of discrete phenotypes. So these discrete phenotypes, they're still persisting together. So that's the cohesion species concept. Then there's the ecological species concept, in which we are looking at set of organisms that exploit a single niche. They're thought of as belonging to the same species. So if there is a set of organisms and all of them are exploiting the exact same niche, or they are occupying the same adaptive zone, the exact same one, then they will can be defined as belonging to the same species. So that's the ecological species concepts. You're trying to fill in the ecological niches, and there is a competition for resources. And then there's the evolutional species concept, as the term suggests, in which we're looking at the evolutionary lineages to describe a species. And there is this pluralistic species concept, in which you're using a combination of multiple of the above species concepts as appropriate to your particular example. So these are the different species concepts. Now, coming to the ones that are in our focus, number one is the morphological species concept, which was commonly applied in old times when we did not have access to say genome databases or a way of sequencing the genome, and we did not know enough about the behavioral the reproductive behavior of the species. That time, or when naturalists just begun, they used to describe species based on their morphology, and members of a species should be morphologically similar to each other. The basic idea behind morphological species concept is that members belonging to the same species should look similar, they should be morphologically similar. So they are different in certain fixed characteristics. So there is a fixed character difference, and it entirely relies on morphology. Hence, it is easier to apply it to extinct species for which we only have fossil evidences available. But it comes with its own caveats, as if you're talking about species, we now know that there are species that are sexually dimorphic, in which males and females look different. There are so many birds, for example, the paradise flycatcher, the peacock, the jungle fowl, pheasants. There are so many birds that are actually sexually dimorphic. Males and females look very different from each other, and naturalists can very easily end up defining them as different species. For example, you have this image of a butterfly species here, in which the males and females look so distinct that they can be easily classified as two different species if the naturalist is not actually following them and looking at their breeding ecology. There are spiders also, in which there is females that are 10 times bigger than the males, and they look very distinct from the males, and anybody would think of them as belonging to a separate species if they were not aware about their behavioral ecology, or about their reproductive behavior, right? So this is where the caveat comes in of morphological species concept, that you cannot use it everywhere unless you are 100% sure and completely aware about its breeding ecology as well. So it is not so again, morphological species concept is also not foolproof. You cannot just describe a species based on its morphology, or based on its physical features, correct? Then eventually Ernst Mayr, he proposed the biological species concept, in which what he proposed was that a group of potentially, actually, or potentially interbreeding natural populations that are reproductively isolated from each other or from other such groups are called species. So it's a group of actually or potentially interbreeding individuals and or natural populations, and they're reproductively isolated from other such groups. From other populations, they're reproductive isolated. So from the other groups from which they are reproductively isolated, they are separate species from them. But within the species, the populations can potentially interbreed. Why are we using the term potentially? We're using the term potentially because these populations, they might also be separated from each other in space, and they might not be actively interbreeding with the organisms of the other population, but if put together in the same space, they would still have the potential to interbreed and produce a fertile offspring, right? So that is why the term actually or potentially interbreeding natural populations. So when you're talking about such a group of actually or potentially interbreeding natural populations, and they are reproductively isolated from other such groups, then these are thought to belong to a similar species. So, in simpler words, individuals that are interfertile, they belong to the same species. So, if there are individuals that can breed with each other, that can produce a fertile offspring, they are called said to belong to a same species. And for a biological species concept, you are using or uh, the biological species concept actually brings in the concept of reproductive isolation in defining a species. And according to this concept, individuals of different populations may not mate with each other, or their mating may not result to produce an offspring, or the offspring that is produced may not be viable or fertile. And if any of this case is applicable, then those individuals are said to belong to different species. So the biological species concept, as I already said, brings in the picture the concept of reproductive isolation for defining a species. And it says that if individuals that are belonging to different populations, if they are not able to mate with each other, if they can mate but they are not able to produce an offspring, if they are producing the offspring but the offspring is not viable or it's not fertile, then they're said to belong to different species. Again, biological species concept comes with its own caveats. First of all, since we are very strongly talking about reproduction, it cannot be applied to any of the asexual organisms, and we know that so much or much more of the biomass of the earth's living organisms is occupied by these asexual organisms, which are bacteria, viruses, all of them. So it cannot be applied to any of these asexual organisms which divide by things like binary fission. So that is a major caveat of the biological species concept. It cannot be applied to extinct species for which we do not have detailed reproductive information. Also, to use biological species concept even on extinct species, you need to have a very detailed um, information about their reproductive biology, about their behavioral ecology. And if that species is in a remote location and you're unable to get to that reproductive biology information, you might not be able to apply biological species concept to define that species. And it is difficult to apply to species which have a complex reproductive biology. So if a species has a complicated reproductive biology, again BSc becomes difficult to apply to such species. For example, if you're talking about species, there are certain species that follow a ring distribution. So imagine that there is this certain mountain range which is a barrier between certain species. Then what a ring species are is that they are the same species, or the populations of the same species follow a ring pattern. So the organisms that are found here and the organisms that are found here, which are so geographically separate from each other, which are geographically so far away from each other, they belong to the same species. And why is that? Is because there are multiple populations of the same species present in a ring like manner around the obstacle or around the geographic barrier. And all of these populations actually maintain interbreeding with their immediately next populations. So if you're talking about this population, it will be able to interbreed with the immediate neighbors on both sides, but they will not be able to interbreed with populations here or the populations here. So this is the definition of a ring species. And to these complex reproductive biologies, it is again difficult to apply the biological species concept. Okay, so yeah. So as I was saying, for the biological species concept, it's difficult to apply it to species with complex reproductive biology. And a ring species is a species in which the populations of the species are present all around a geographic barrier. But the adjacent populations can interbreed, but the populations at the very far end actually cannot interbreed with each other, and it leads to a gradual change into speciation. Coming to the phylogenetic species concept, as the term suggests, the phylogenetic species concept uh, uses phylogeny or uses evolutionary lineages to define species. And it says that a species is the smallest diagnosable monophyletic group. So, a monophyletic group is the one which shares one common ancestor. So if you have these different clades, right, then under this one ancestor, all of these are called monophyletic because they belong to one common ancestor. But if this particular or species is also related to these species, sorry, if this particular individual is also related to these individuals, then that is not monophyletic because it has a separate ancestor as compared to these ones. So the smallest diagnosable monophyletic group, that is how a phylogenetic species concept defines a species. And it defines a species as a group of organisms which share a pattern of ancestry and which share their descent, and they form a single branch on the tree of life. So a species is a single branch on a tree of life. So this particular uh, branch here is one species. This branch here is one separate species. So that is how the phylogenetic species concept defines a species. It's a group of organisms that share a pattern of ancestry and descent, and they form a single branch on the tree of life. Now, why do I have the example of old world vultures and the new world vultures? So even though both of these groups are actually called vultures and they have behaviors matching the definition of being a vulture, but when scientists actually looked into their phylogeny, they found that the old world vultures are much closely related to eagles and kites, but the new world vultures are actually much more closely related to storks. So that is how the phylogenetic species concepts gives us insights into different kinds of organisms and their phylogenies, and it describes species based on their evolutionary history and their ancestry and descent, and not on their reproductive isolation or morphology. Now, and the last species concept that I would like to talk about is the lineage or the general species concept. It was actually put forth by De earlier in 1998 and then again in 2007. In this, they were actually talked about a lineage as an ancestor descendant species, or a series that is descendant from an ancestor, in this case of metapopulations, or simply a metapopulation. Yeah. So De put forth the De put forth the lineage or the general species concept, in which a lineage actually refers to an ancestor descendant series, and the metapopulations, or simply a metapopulation which is being extended through time. So you might not be aware of what a metapopulation is, but a metapopulation is actually a collection of multiple populations belonging to the same species. Okay. So, De was talking about in the generalized linear species concept is that you can apply multiple species concepts, and these multiple species concepts can happen at different in different combinations and at different times. So, for example, here in this image, you can see that when it's an ancestor, there's clearly one single species, and then there is a gray zone where you don't know whether to call them two different species or not, and then there is a time after which you can actually call them two different species. And then in this gray zone, according to the lineage or the general species concept, you can actually call them different species, because even if one of the species concepts is correct, then you should be able to call this species two different species, even if one of these different species concepts, say behavior, ecological, or biological, phylogenetic, even if one of them is applicable, you should be able to call them different species. So that is what the general species concept is all about, that there is a one ancestral species, and then there is a certain gray zone in which some of these species concepts are applicable but others might not be, and then eventually when most of these species concepts are applicable, you start calling them two different species. But the moment when one of these species concepts is actually applicable to this species, you should be able to distinct them into two different species, right? Under all the species concepts, a species is separately evolving metapopulation lineage. So when you're plotting these metapopulations on a phylogenetic tree, if they're falling into different lineages, even under one of the species concepts, then you should be able to call them a separate species, right? So moving forth, so because we discussed about reproductive isolation very majorly in the biological species concept, we should know what actually reproductive isolation is. And as the term suggests, reproductive isolation means when individuals are isolated from each other, they're unable to breed, they're unable to mate. So they're said to be reproductively isolated from each other because they cannot produce a fertile offspring, right? That is what reproductive isolation is all about, and there are different stages in reproductive isolation. First of all, there is a reproductive isolation which is pre-mating. As the term suggests, it acts even before these individuals, it acts at a level which prevents the individuals from mating with each other, right? It could be behavioral, could be temporal or seasonal, mechanical or ecological or habitat. Behavioral, as the term suggests, behavioral pre-mating isolation happens when individuals do not recognize each others as their mates, right? For example, you might be aware of bird calls, frog calls, cricket calls. In all of these, individuals calling or vocalization is a method in which they attract mate, in which they choose mates. Now, if there are populations which are even slightly different from each other in their calls, the individuals might not be able to recognize their counterparts, their countermates, and they might not breed with each other. So that is one way of isolation, which is behavioral isolation. Temporal or seasonal isolation means when individuals they reach their maturation in different seasons or in different times. For example, say there is a cricket species which actually breeds in summer, and there is another cricket species which actually breeds in monsoon. For example, say species one of the cricket species breeds in, or the population one breeds in the months of May to June, and population two breeds in August to September. Then these both populations, even though they can potentially or even though they can produce even though if you fertilize their gametes separately, they might be able to produce offsprings, but since they are temporally separated from each other, naturally they will never end up breeding and producing offsprings, right? So that is another way of pre-mating isolation. Then there is mechanical isolation, in which the reproductive parts have so evolved that they do not no longer fit into each other. For example, the reproductive parts actually follow a lock and key mechanism, in which the male reproductive parts fit into the female reproductive parts, and that is how mating happens. But when they're mechanically isolated, then these two parts actually do not are unable to fit together, and they're unable to uh, mate. The individuals are unable to mate if they belong to these populations in which are mechanically isolated. Ecological or habitat isolation is that if there is one population that lives in ecosystem A and another population in ecosystem B, and if these ecosystems are so evolved, they're so changed that the populations do not exit these ecosystems, then they'll never be able to meet each other, and they'll never be able to carry out breeding. So again, these populations are reproductively isolated even before mating can happen. So in all of these mechanisms, behavioral, temporal, mechanical, and ecological, mating itself is not happening. So that is why it's pre-mating reproductive isolation. If you're talking about post-mating pre-zygotic reproductive isolation, in this mating has actually happened, but the zygote is unable to form. So that is why it is called post-mating pre-zygotic isolation. So the mating has happened, but the zygote is unable to form, right? So two types in which this can happen is number one, conspecific sperm precedence, in which even if individuals belonging to different species they try and mate, but the precedence in the female body is given to the conspecific sperms, or to the sperms belonging to males of the same species, and that is why the sperm of the other individual, which either belongs to a different species or a different subspecies, is unable to fertilize the ova of the female belonging to this species A, right? So that is how conspecific sperm precedence takes care of post-mating prezygotic isolation. Then there is gamete isolation, in which even though they are able, the individuals have mated and the gametes have been exchanged, but there might be certain cell surface proteins which make them incompatible, which makes it impossible for the two uh, cells to actually fuse together, for the ova and the sperm to actually fuse together and produce a viable zygote. So that is why it's a prezygotic isolation, in which the zygote is unable to form. Then the last but not the least is actually the post-zygotic isolation, in which the reproductive isolation happens at the level of in which mating has happened, zygote has formed, but now that zygote might not be able to give rise to a viable hybrid, or it might give rise to a sterile hybrid. And that is why even though the hybrid has formed, there might be certain problems with the hybrid which makes them still reproductively isolated. So the hybrid that's formed by such a mating, it might be inviable, that is it's not able to survive. It might be sterile, that is it's not able to give rise to further offspring. For example, a mule which is formed by a horse and a donkey, it is sterile, it is not mules are not able to interbreed among themselves. And there could be a hybrid breakdown, in which the first filial generation is correctly formed. For example, the mule is for example, just imagine that species A and species B hybridize and they form a hybrid, but these hybrids now they themselves cannot give rise to a viable second filial generation. So the F2 is not viable. That is when it's called hybrid breakdown. Okay, so these are different forms of reproductive isolation that happens. Moving on to speciation, how speciation, or what is speciation? Speciation, as the term suggests, is actually a process in which a lineage splits and results into two daughter lineages, in which there is one ancestral lineage and it splits and results into the formation of two daughter lineages. For example, there is an ancestral species and certain events happen and it gives rise to, or the two populations give rise to two different species. That is what is a speciation. So it's a process of lineage splitting that results in divergence of ancestral lineage into daughter lineages. And uh, that is what is speciation. And the major causes of speciation is a barrier to gene flow. When there are multiple populations but they're unable to exchange their genes among themselves, then they become genetically isolated and and they then split into two daughter lineages. Types of speciation can be allopatric, parapatric, peripatric, and sympatric. These are certain types of speciations. Here you can see in this diagram how these speciations are very well described. In an allopatric speciation, you have an original population. Then initial step in the process of speciation is that there is a formation of a barrier in this population. And then eventually, because of this barrier, because there is a barrier to the gene flow, there is evolution of reproductive isolation, that the population on the left and the population on the right are unable to um, reproductively or there is no gene flow, they're reproductively isolated, they're unable to interbreed. And now, even if the barrier is taken away, these populations have formed into distinct species, and they will not be able to interbreed. Right? That is what is allopatric speciation. Then there is a parapatric speciation, in which there is an original population. Then this original population, a small subset of the population, enters a new niche. This new niche that is entered now leads to the development of certain traits which are only present in this small subpopulation, and these might get again reproductively isolated. So in this new niche, all the individuals present in the new niche, they might be reproductively isolated from the other individuals present in the original population. And now, even if these individuals from the new niche come back to the original population, they will still be unable to interbreed with the individuals of the original population. So that is how parapatric speciation works. That some of the individuals from the original population enter into a new niche and they develop certain characteristics, certain traits that makes them reproductively isolated from the original population. In sympatric, there is an original population. In this original population, for example, certain polymorphism occurs and they have certain different traits, and now these individuals are unable to interbreed with the other individuals of the population and they can only breed among themselves. So within the population, this trait spreads, and the individuals that have this particular trait, they are unable to breed with the individuals of the rest, or the uh, with the rest of the uh, population, with the original rest of the original population. And there is another speciation which is called a peripatric speciation, in which what happens is that similar to parapatric, there is an original population and there is a separate zone here, and the so it's not a new niche, but it's a new habitat which is a little away from the habitat of the previous population. And individuals from this population, they enter into this new habitat and they occupy this new habitat and they become reproductively isolated from the parent population. And that is what really is peripatric uh, speciation. So in parapatric speciation, the new niche that is entered is actually still connected to the previous population. But in peripatric speciation, this is peripatric. In peripatric speciation, the new niche or the new habitat that is entered is actually isolated. So there is no movement between these two. So that is actually isolated, and that is how peripatric speciation takes place. Okay, so there's a very slight difference between peripatric and parapatric. Peripatric is more like allopatric and parapatric is more like sympatric. Okay. Coming to describe in more detail what is allopatric speciation. So there is an initial separation between between an original population due to the development of certain geographical barrier. For example, say there is a population and now suddenly a river changes its course and it starts flowing between these two between this population, and it separates them into two different populations. And this is eventually followed by divergent selection for different traits, for different niches. And if these two areas have different environments, then these two populations will develop certain different traits, and eventually there will be two different species. Even if you place them in a similar in in a common area, they'll still not be able to reproduce, they'll not be able to breed, and that is how they'll be reproductively isolated. So there is a genetic divergence due to barrier to gene flow. There was a barrier to the gene flow between the two separated populations, and this has led to genetic divergence, right? So that is allopatric speciation. Coming to sympatric speciation, speciation that has happened within the geographical region due to certain intraspecific competition or due to certain polymorphism that has come up into the population and has gotten fixed. And this genetic variation of the polymorphism gets fixed into the population, and these individuals that have this genetic variation, they're unable to interbreed, or they're unable to breed with with the individuals belonging to the original species, right? Now, what is biodiversity? So, biodiversity, now that we've discussed species and we've discussed speciation and how speciation occurs, what are the different barriers, now let's talk about biodiversity. Biodiversity is the variety of life in all its forms and at all levels of organization. So, when you're talking about variety, biodiversity is when you're talking about variety of life. It can happen in all the forms of life and at all levels of organization. And what are different levels of organization of biodiversity that you can think of? One is genetic biodiversity. So, in which within a species, different populations are genetically diverse. So, even we as a species, we are genetically different from our friends, from our family as well. So all of us are genetically distinct from every other organism around us. That is genetic biodiversity. Species biodiversity, within a certain ecosystem, so you can look around you, there are many different species. So this variety of species present within a particular ecosystem is called ecosystem species biodiversity. Then there is ecosystem biodiversity, in which case you're talking about different ecosystems present on this planet Earth. So that leads to an ecosystem level biodiversity. And one last biodiversity that I have forgotten to list here is actually functional biodiversity, in which we are talking about the biodiversity of the different functional roles that are present within an ecosystem. And we'll come to it later. So there are these different types of different levels of biodiversity. Coming to the basic one, the genetic level of biodiversity, the biodiversity that is present in the genetic information at various taxonomic levels. So it can be measured in two ways. Number one is polymorphism. Polymorphism is actually the proportion of alleles that are represented in a population which have more than one alleles. So it's the proportion of genes that have more than one alleles, right? For example, if you're talking about height, there might be alleles like tall, medium, and short. There might be, if there is color, there might be alleles that say that the eye color can be brown, blue, green, black, anything like that. I'm taking an example in humans. So that is polymorphism. There is a single gene, so there is a trait, for example eye color, and it is represented in a population by more than one alleles. There are different alleles controlling different colors of eyes that we have. For example, as I said, green, blue, brown, black, like that. So polymorphism is the proportion of genes that are represented in a population by more than one allele. So what is the proportion of genes that have more than one alleles in them, right? Then there is something called heterozygosity. Heterozygosity is the proportion of genes at which an average individual is heterozygous. So, if you're talking about one average individual in that species, or for example, if you've looked at all the individuals in a species and you take the average of the proportion of genes at which an individual is heterozygous. For example, you could be homozygous at a particular gene, as we have discussed. You might have, if you're talking about a gene which is and you for example, you're talking about a gene for eye color. If both your alleles are blue and blue, then you are a homozygous. If your allele is blue and green, if your allele is blue and brown, then you are a heterozygous. So what is the proportion of genes at which the individual is heterozygous? That is actually heterozygosity. Okay, this is genetic diversity. Another way to talk about genetic diversity is the presence of different number of alleles with different distribution frequencies in a certain population. So we're talking about that for certain genes, the number of alleles is more than one. So there is a various number of alleles and they have different distribution frequencies in the population, right? And genetic variation simply refers to the variation of genomes at individual level or at species level. So you're talking about variation of genomes either at individual level or at species level between species populations, right? Right. Talking about species diversity, so when you look at species diversity, you're looking at how in an area or across areas, what is the species diversity, what is the different types of species present in that particular ecosystem. So there are different ways of measuring species diversity. Here I've put um, here this image shows there is alpha diversity, in which if you're talking about one particular region, what is the species diversity within that particular region, that is shown by an alpha diversity. Here you can see that in this circle there are three different species of plants. There are trees belonging to the species A, B, and C. So that is the alpha diversity. There are three different plant species. Beta diversity is when you compare two different areas and when you compare species between these two areas, that is beta diversity. For example, if you're comparing species in this area and this area, then that is beta diversity. And if you're looking at a large area and you're looking at the species diversity at that large area, or you're comparing different multiple smaller areas for their species diversity, that is called gamma diversity. The alpha biodiversity. I'm sorry, those two slides. We were interrupted by the importance and the uh, of the biodiversity. Now let's get back to how to measure alpha biodiversity. So there are these two indices. There is the Simpson's diversity index and the Shannon diversity index, which helps you calculate alpha biodiversity. In Simpson's diversity index, for calculating biodiversity, D, or the diversity, is equal to 1 divided by the summation of pi square, from I is equal to 1 to S. So here pi is the proportion of individuals belonging to that particular species. Pi is equal to n over capital N, where n is the number of individuals belonging to this species and capital N is the total number of individuals in the area. S is the total number of species in the area, right? So that is so the Shannon Simpson's diversity index is D is equal to 1 over summation pi square, I going from 1 to S. And Shannon diversity index H is equal to the negative summation of pi ln pi, and summation goes from I is equal to 1 to S again. Pi again is the proportion of individuals belonging to that species, and S is the total number of species present in that area, right? Okay. So these are the formulae for Simpson's diversity index and Shannon diversity index. Now we have the small examples. So we can solve these examples. So area one, first example, in area one, it was sampled and the following specimens were collected. So you have grasshopper, butterfly, butterfly, and beetle. These were the specimens that were collected. So to describe, there was a grasshopper with which was green with red legs, one which was brown with yellow stripes, one was large and blue butterfly, one was a small and blue butterfly, and one was a red and blue beetle. So the number of individuals, small n, is 6, 5, 1, 3, and 12. Now if you want to calculate, so let's see how we calculate this, uh, right? So I will give you a brief example on how to calculate the alpha diversity for this area 1. For this I will open my Excel sheet, right? You can also open your Excel sheets and copy the contents of this area 1, in which we have this description and species. So I will just write these as species 1, 2, 3, 4, 5. We have 5 different species in this area 1. So yeah, so let me share my screen with you. Here you'll be able to see that we have different species. Species, say we have A, B, C, D, and E. A number of individuals, small n, here we know is was how much? 6, 5, 1, 3, and 12. There are 6 individuals, 5 individuals, 1, 3, and 12. So capital N, which is the total number of individuals in that area, is actually the sum of these different individuals. So it's actually 27. So when you come to proportion of individuals of that particular area, which is pi, that is equal to 6 divided by, this is equal to this cell divided by, what is the total number? 27, right? So this is the proportion, and this is the proportion for the other ones. And if you're actually calculating the the Shannon diversity index. In this, what you want is you have pi. Now you want the natural log of pi, right? So in this, you will take the natural log of this number. So this is the natural log of numbers. And what we want is we want the summation of pi, ln pi. So here we will multiply pi with the natural log of pi, right? So it is equal to this multiplied by this, and this is what you will get. And your answer here will be, so the answer here will be negative of this number. So 1.37. That will be your Shannon diversity index for the area A, right? So I hope that was easy for you guys to follow, and we'll get back to our slides now, and you can follow. You can look at the slides again and follow the same methods to calculate the diversity index of of the area too, right? So again, a solution here is given. So if you calculate H, which I did, which is the Shannon diversity index, you get 1.37 as we got. And if you calculate D, which is the Simpson's diversity index, you get an answer of 3.4. So here is how we calculated it. We have n by n, which gives you the proportion. We have pi square, and then we have log pi, npi, ln pi. And we used this formula. We did not use the pi square formula, right? So you calculate all of this, and to calculate evenness, you divide d by d max. D max for this area would be 5 because we have 1, 2, 3, 4, 5 species, right? So that is how you calculate the evenness from these two indices. Again, for species 2, as you might have expected, since we have more number of species, so we'll get a higher diversity indices for these two species. So you can go through these slides, and you can try and calculate these for your own self to understand how this formula is working and how we are calculating the alpha diversity of an area. Moving on to beta diversity, we have different indices, Jacquard's index and Sorensen's index. In the Jacquard's, so for beta diversity, what you want to keep in mind is that, say you have two different areas, A and B, and in those areas you have different number of species. For example, say small a is the number of species present in area A, small b is the number of species present in area B, and small c is the number of species that is common to area A and B. So for Jacquard's index of beta diversity, Sj, it will be C divided by A plus B minus C, which is the number of common species divided by the total number of species in both the areas. And for Sorensen's index, you do is 2C divided by A plus B, which is the number of common, twice the number of common species divided by the A plus B, which is total number of species in the two areas. Counting the common species twice actually. And when you look at the dissimilarity indices, you basically just subtract both of the similarity indices from one, and you get the dissimilarity between these two areas. Again, we have an example here, so you can calculate the similarity between sites B and C in this example and see what you get as an answer, and you can solve it for your own self, right? So mostly for this Jacquard and Sorensen's index, you actually just look at present absence data, so you do not need to take care about this 22, 14, and all of this. If there are certain individuals, you just take them as 1, and if there is nothing, you take that as 0. Moving forward to the species accumulation curve. What is a species accumulation curve? It's actually a graph of the cumulative number of species recorded in a particular environment. as a function of the cumulative sampling effort. So on the x-axis, you have the cumulative sampling effort. Here it is the cumulative number of plots that were sampled. Y-axis, you have the cumulative number of species found. So if in the first plot you sampled and you saw one species each, so you put one. Then if so there are two of these curves. One of them is with the brown with the open dots, one of them with the solid dots. How these two are different, we'll come to it later. So if you come to one plot, so you sampled it, you saw one species, then you move to another plot, you saw another. So here now you have seen two species. So you put a dot at two. A total of three species, and then you only see three species. You don't see anything new. You only see those three species again and again. So this is an adequate sampling because your graph is leveled off and you reached a asymptote. But if your graph does not level off, it does not become like a flat line, then that means that your sampling effort is inadequate and you've not yet reached an asymptote and you should carry out more sampling because you've not yet seen all the species in that area, right? Coming to ecosystem biodiversity, when you talk about ecosystem biodiversity, it is the variety of ecosystems that are found in a particular region and how they are and what is their distribution, right? So the variety of ecosystems found in a particular area and what is their distribution. For example, in this diagram you see that there are many different ecosystems. There is this grassland, there is natural forests, there are artificial forests, there are kapais, there is irrigation ponds, there is these leaves, there is rice paddies. So there are so many different ecosystems in that particular area itself. So there is grassland, forest, and agro ecosystem as three bigger brackets of the multiple ecosystems found in this area. That is ecosystem biodiversity. How many different types of ecosystems you have in a particular area, right? Then ecosystem biodiversity addresses the combined characteristic of biotic properties and abiotic properties. Because in an ecosystem, when you talk about an ecosystem, you have species, which are the biotic properties, and also the abiotic properties like the temperature and the humidity and the landscape properties, right? So when talking about ecosystem biodiversity, it addresses what are the combined factors of biotic properties and abiotic properties, and it is a variation in ecosystems found in a region or the variation in ecosystems over the whole planet. So you might be talking about one particular region, for example, we did in that particular diagram. I might be talking about the whole planet. And ecological diversity includes variation in both terrestrial and aquatic ecosystems. And you have these different ecosystems like the tundra, the desert, the forests, the grasslands, the savannas, which are the grasslands themselves, and the oceans. There are these many different ecosystems present on the face of the earth. And when you come to functional ecosystem, functional biodiversity, it's actually when you're talking about what are the range of traits of organisms that are present in a community. So when you're looking at the community, what is the range of different traits of organisms which are present in that particular community? For example, as we discussed already, there might be some nitrogen fixers, there might be predators, there might be herbivores, there might be decomposers, autotrophs, all of these different um, things might be present. So when you're talking about functional biodiversity, then when you're talking about what are the different traits of organisms present in the community, right? Now let's come to what is the importance of biodiversity. Why do we need biodiversity? So eventually I'll come talk about ecosystem biodiversity and functional biodiversity as well, but it's important for us to also know why is biodiversity important. So it offers us food security. So there are plants, animals, and mushrooms. In an area which is more biodiverse, we have more options of plants, animals, and different types of things that we can consume. Economic benefits, you have foods, you have plants and animal products, you get medicines. So an area which is more biodiverse can actually give you more of these economic benefits. Then there are these biological control agents. For example, you have diseases and invasive species. So if you have an area which is properly biodiverse, which is well biodiverse, then you will notice that in those areas the incidence of disease is much lower, and the probability of an invasive species setting up in that area is also much lower because that area is fully biodiverse and there are no niches left for that invasive species to suddenly start and occupy. A highly biodiverse areas also offers you a lot of ecosystem services, for example, pollination services, fertilizers, soil irrigations, and things like that. And it has better aesthetic values because nature is very calming to look at. It's very soothing, and it offers you different benefits. Even though we don't realize these are very important for our mental and physical well-being. And then there are these certain scientific values that come with biodiversity. For example, we can look at these evolutionary science, anatomy, physiology. If we don't understand the anatomy and physiology of different organisms, we'll not be able to understand the anatomy and physiology of human beings themselves. So before applying anything to human beings, you know that all of these medicinal trials are applied to other individuals, other species. That is again how biodiversity is important for us. Now, diversity also brings about stability. How is that happening? How is diversity bringing in stability? So when you have a lot of genetic diversity, you see that these population will be, at least some of the individuals in the population will be disease resistant, some of them will be drought tolerant. So if you're talking about certain populations, then if you're talking about them being genetically diverse, then you'll see that they'll be resistant to a lot of these unfavorable conditions, like their disease spread will not happen in such a population which is genetically diverse. They might be drought tolerant. Some of these species might be tolerant to higher water levels. Some of them might be tolerant to higher salt levels. All of these things might be happening. Species diversity, if you have a lot of species, for example, insects, cats, dogs, and humans, you need a lot of species to keep everything in a well balance. And functional diversity, again, you require a lot of functional diversity to keep all the ecosystem properly functioning, because you require the apex predators, mesopredators, nitrogen fixers, everything to be present for a species, for an ecosystem to be working properly. For example, take the example of the Yellowstone National Park. In the Yellowstone National Park, they actually the humans actually erased, or they decreased the number of the wolves entirely. And that was because they did not want these wolves to be there, because they wanted the elk populations to survive, because they were hunting these elk populations. Also, they were scared. Humans were scared of these wolves and they thought they were very dangerous. So they actually hunted all of them down and they drove them away from that population. And they wanted those grasslands to develop properly. There were so many sheep herders that were using the Yellowstone National Park grasslands as their herding grounds, and they didn't want the wolves to be feeding on their sheep populations, and so there was another reason to drive away the wolves. Then what was seen was everything went haywire in the Yellowstone National Park. A lot of the elk populations shot up, and they were consuming all of these new saplings, these grasses, everything, and still the sheep was not getting enough food, and the quality of the habitat had gone down much. It was much worse, and the rivers were changing course, and everything was going haywire until they actually introduced the wolves back again, which controlled the elk populations, which gave rise to more trees and more grasses, which were able to hold the soil together, preventing soil erosion, and the rivers were not able to meander so much, and they also had their steady courses. So even one single functional guild can influence the entire ecosystem. Now, you might already be aware of what are biodiversity hotspots. But biodiversity hotspots are regions in which there is high level of species richness, there is a high degree of endemism, and there is a high degree of threat, or the species found here are actually vulnerable. And in India, we have these four different biodiversity hotspots. There are the Himalayas, there are the Indo-Burma area, there is the Western Ghats in Sri Lanka, and there's the Sundaland in the Andaman Nicobar Islands, right? So these are the biodiversity hotspots. This brings us to the question and answer session for today. This was all about what was discussed during this week and a little bit extra information on what was discussed. I would actually like for you to go through the beta and the alpha biodiversity indices and try and calculate the biodiversities for these different zones and see how it comes out. Or if you want an example, I can actually calculate the biodiversity of this for you. See, give me a second. We can actually, I can again, okay. So I can write this information down for you guys. So we have 10 species. Out of these 10 species, in site A, B, and C. So let's see, we have species, we have site A and site B and site C. And we have 10 species, right? And of these, in species, in in the site A, we have 1, 2, 3, 4, 5, 6, 7, 8, and 9 species. In species A, we in area A we have actually 9 species. In site B, we have 1, 2, 3, 4, 5, 6, 7, 8. Right? We have 8 species in the site B, and in site C we have 1, 2, 3, 4, 5, 6, 7 Right? We have 7 species in site C, and since we are only comparing site B and site C, let's see how many common species we have in these two areas. So in these two areas, so give me a minute. In areas site B and site C, the common number of species we have 1, 2, 3, 4, 5, and 6. So we have C, or sorry for the bad writing, C is equal to 6, which is the common species in site B and site C. And for site C, the unique species is 1. So total number of species in site C is actually 7, and total number of species in site B is actually 8, out of which 6 species are common. So if you come back to so Ranson's index, you do 2c over a plus b, so which means 12 divided by 8 plus 7. That will be your answer. 12 divided by 8 plus 7, 15. This will be your 4 by 5, which will give you the Sorensen's similarity index. Okay, so I hope it's clear now how do you calculate uh, the similarity indices, the beta diversity between two different areas, right? Okay. Now, moving forward to the Q&A session for today, I will ask you some questions. This might help you with your understanding, and it will clear some concepts for you guys if you're able to answer these questions. Okay. Number one, what is speciation? Is it the extinction of a species, the process by which new species arise, the adaptations of individuals to their environments, or the increase in population size? So, what exactly is a speciation? And so, yeah, the correct answer to this question is that speciation is the process by which new species arise, which is option B. Next question, which of the following is not a type of speciation? Allopatric, sympatric, convergent, or peripatric. So we've discussed the different types of speciation during this lecture. So which of these is not a type of speciation? I will give you 10 seconds to think about the answer, and then I will tell you the answer. And so, yes, the correct answer is actually convergent speciation. We've discussed allopatric speciation, in which a geographical barrier comes up and separates a population into two distinct populations, which actually form two new species. Sympatric population, in which within the population there might be a polymorphism of alleles which might develop and might get fixed in the population, which leads to a rise of different species. That is sympatric speciation. And then there is convergent, which is not a type of speciation. So we cannot, so speciation convergent is actually not a type of speciation. And there is peripatric speciation, in which we discussed that a certain subpopulation enters into a separate niche, which is actually isolated from the rest of the parent population, and this subpopulation which has moved into the new niche now becomes a distinct species in itself. Now, allopatric speciation occurs when two populations evolve into different species due to physical separation. A species becomes extinct. Different species evolve similar traits. Two populations merge into one. So, when is allopatric speciation thought to occur? So, yes, the correct answer to this question is when two populations evolve into different species due to physical separation. That is allopatric speciation, as we've already discussed. I just gave you the answer in the previous question. Okay, reproductive isolation prevents what? Does it prevent genetic drift? Does it prevent gene flow between different species? Does it prevent natural selection? Or does it prevent evolution from occurring? So, what is reproductive isolation preventing? Yeah, so the correct answer to this question is that reproductive isolation prevents gene flow between different species. Which of the following is an example of prezygotic isolation? So, which of the following is an example of a prezygotic isolation? Is it hybrid sterility, hybrid inviability, temporal isolation, or hybrid breakdown? Which of these is an example of prezygotic isolation? So, as you might have already guessed, the correct answer is temporal isolation. All of the other ones, hybrid sterility, hybrid inviability, and hybrid breakdown, are actually examples of post zygotic isolation because the hybrids are either sterile, they are unable to reproduce themselves, they are inviable, they're unable to survive, or the second field generation is unable to reproduce or survive. So that is why these are all post zygotic isolation processes. Only temporal isolation or seasonal isolation is a form of prezygotic reproductive isolation. A mule is an example of what? Is it a hybrid inviability, hybrid sterility, temporal isolation, or behavioral isolation? So, what is mule an example of? You again have 10 seconds to answer this question or think about the answer to this question, and then I will let you know. So the correct answer to this question, as you might have already guessed, is hybrid sterility. Because mules are viable, you do find them living, but they are not able to reproduce and produce offsprings themselves. The biological species concept defines a species based on what? Is it morphological differences or genetic similarities or the ability to interbreed and produce fertile offsprings or the ecological roles in the environment? So, what does the biological species concept use to define a species? Yeah, so the correct answer to this question is that a biological species concept defines a species based on the ability to interbreed and produce fertile offsprings. Which species concept is based on physical characteristics? Is it the biological species concept, morphological species concept, phylogenetic species concept, or the ecological species concept? Which of these following is based on physical characteristics, also called phenotypes? Yeah, so the correct answer to this question is actually the morphological species concept. Phylogenetic species concept classifies species based on what? Their ability to interbreed, their ecological roles, evolutionary history and common ancestry, or the presence of reproductive barriers. So, what does phylogenetic species concept use to classify species? And yeah, so the correct answer to this question is evolutionary history and common ancestry. Moving on to what does biodiversity refer to? Is it the number and variety of species in an ecosystem? Is it the genetic diversity within a single individual? Is it the amount of sunlight available in a habitat or the reproductive rate of a population? So, what does biodiversity refer to? And yeah, so the correct answer is that biodiversity is the number and variety of species in a given ecosystem out of these four options. Which of the following increases biodiversity? Is it habitat destruction, invasive species, conservation efforts in habitat restoration, or climate change? Which of the following is actually a way of increasing biodiversity? So, the correct answer to this question is conservation efforts in habitat restoration. Which ecosystem is known for having the highest biodiversity? Is it a desert, a tropical rainforest, or a tundra or a grassland? Which of these ecosystems is known to have the highest biodiversity compared to the others? And yeah, the correct answer to this question is actually tropical rainforest because these are forests which have high amounts of productivity and so they have a high species diversity. Species richness is a measure of genetic variation within a population, number of different species in an area, total number of individuals in a species, or the physical characteristics of a species. So, what is species richness a measure of? So the correct answer to this question is that a species richness is a measure of the number of different species in an area. What is species evenness? Is it the number of species in a region? Is it the relative abundance of species in a community? Interaction between species or total biomass of an ecosystem? And yeah, so the correct answer to this question is species evenness is the relative abundance of species in a community. Shannon index and Simpson index are used to measure what? Species extinction rates, genetic diversity within a population, biodiversity by considering species richness and evenness, or the number of predators in an ecosystem. This is the last question for today. So let's see what Shannon index and Simpson index are actually used to measure. As you might have already guessed, the correct answer to this question is that they're used to measure the biodiversity by considering both species richness and species evenness. So, this was all for today. I hope this was an informational lecture. And if you have any doubts, you can type them out in the YouTube comment section, or you can either type them out in your discussion forum in NPTEL. And if you have any further doubts, you can also reach out to me. And yeah, you can find these. So the link to finding these slides will be present in the YouTube video itself. Okay. Thank you.